Good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:15 a.m. Eastern Time. Quick note. Scientists find evidence of ancient mega drought in southwestern U.S. Science Daily, November 6, 2011. A new study at the University of Arizona Laboratory of Tree Ring Research has revealed a previously unknown multi-decade drought period in the 2nd century A.D. The findings give evidence that extended periods of aridity have occurred at intervals throughout our past. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:16 a.m. Eastern Time. Quick note. Almost 900 years ago, in the mid-12th century, the southwestern U.S. was in the middle of a multi-decade mega-drought. It was the most recent extended period of severe drought known for this region. But it was not the first. The 2nd century A.D. saw an extended dry period of more than 100 years, characterized by the multi-decade drought lasting nearly 50 years, says a new study from the scientists at the University of Arizona. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:20 a.m. Eastern Time. Quick note. Dendrochronology is a precise science of using annual growth rings of trees to understand climate in the past. Because trees abnormally clearly define growth ring around their trunk each year, counting the rings backwards from a tree's bark allows scientists to determine not only the age of the tree, but which years were good for growth and which years were more difficult. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:20 a.m. Eastern Time. Quick note. If it is a wet year, they grow a wide ring, and if it is a dry year, they grow a narrow ring. If you average that pattern across trees in the region, you can develop a chronology that shows what year were drier or wetter for that particular region. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:21 a.m. Eastern Time. Quick note. Darker wood referred to as late wood because it develops in the later part of the year at the end of the growing season. Forms a usually distinct boundary between one ring and the next. The late wood is darker because growth at the end of the growing season has slowed, and the cells are more compact. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:23 a.m. Eastern Time. Quick note. Literally nothing is older than a brisk-cone pine tree, the oldest and longest living species on the planet. These pine trees normally are found clinging to bare rock landscapes of alpine or near-alpine mountain slopes. The trees, the oldest of which are more than 4,000 years old, are capable of withstanding extreme drought conditions. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:24 a.m. Eastern Time. Quick note. To sample the trees without damaging them, the dendrochronologists use a tool like a metal screw that bores a tiny hole in the trunk of the tree and allows them to extract a sample called a core. We take a piece of wood about the size and shape of a pencil from a tree. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:25 a.m. Eastern Time. Quick note. We also sampled dead wood that was lying about the land. We took our samples back to the lab where we used a visual graphic technique to match where the annual growth patterns of the living trees overlap with the patterns in the dead wood. Once we have the pattern matched, we measure the rings and average these values to generate a site chronology. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:26 a.m. Eastern Time. Quick note. In our chronology for the South San Juan Mountains, we created a record that extends back to 2,200 years. It was pretty profound that we were able to get back that far. The chronology extends many years earlier than the medieval period, during which two major drought events in that region already were known from previous chronologies. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:27 a.m. Eastern Time. Quick note. The medieval period extends roughly from 800 to 1300 AD. During that period, there was a lot of evidence from previous studies for increased aridity, in particular two major droughts, one in the middle of the 12th century and one at the end of the 13th century. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:28 a.m. Eastern Time. Quick note. Very few records are long enough to assess the global conditions associated with these two periods of southwestern aridity, and the available records have uncertainties. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:29 a.m. Eastern Time. Quick note. There was another period of increased aridity even earlier. This new record shows that in addition to known droughts from the medieval period, there is also evidence for an earlier mega-drought during the 2nd century AD. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:30 a.m. Eastern Time. Quick note. What we can see from our record is that it was a period of basically 50 consecutive years of below average growth. And that's within a much broader period that extends from around 124 AD to 210 AD, about a 100-year-long period of dry conditions. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:31 a.m. Eastern Time. Quick note. We are showing that there are multiple extreme drought events that happened during our past in this region. These mega droughts lasted for decades, which is much longer than our current drought, and the climatic events behind these previous dry periods are really similar to what we are experiencing today. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:33 a.m. Eastern Time. Quick note. The prolonged drought in the 12th century and the newly discovered event in the 2nd century AD may have been influenced by a warmer than average northern hemisphere temperature. The limited records indicated there may have been similar La Nina-like background conditions in the tropical Pacific Ocean, which are known to influence modern drought during the two periods. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:35 a.m. Eastern Time. Quick note. Although natural climatic variation has led to an extended dry period in the southwestern U.S., in the past there is a reason to believe that the human-driven climatic change will increase the frequency of extreme droughts in the future. In other words, we should expect similar multi-decade droughts in the future, predicted to be even warmer than the past. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 5:50 p.m. Eastern Time. Quick note. Erasing signs of aging in human cells, now a reality. Science Daily, November 3, 2011. Scientists have recently succeeded in rejuvenating cells from elderly donors aged over 100. These old cells were reprogrammed in vitro to induce pluripotent stem cells, iPSC, into rejuvenated and human embryonic stem cells, HESC. Cells of all types can again be differentiated after this genuine rejuvenation therapy. The results represent significant progress for research into iPSC cells and a further step forward for regenerative medicine. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 5:53 p.m. Eastern Time. Quick note. Human embryonic stem cells, HESC, are undifferentiated multiple function cells. They can divide and form all types of differentiated adult cells in the body, neurons, cardiac cells, skin cells, liver cells, etc. Since 2007, a handful of research teams across the world have been capable of reprogramming human adult cells into induced pluripotent cells, iPSC, which have similar characteristics and potential to human embryonic stem cells, HESC. This kind of reprogramming makes it possible to reform all human cell types without the ethical restrictions related to using embryonic stem cells. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 5:56 p.m. Eastern Time. Quick note. Until now, research results demonstrated that since the final stage of cellular aging was an obstacle blocking the use of the technique for therapeutical applications in elderly patients. And his team have overcome this obstacle. The researchers have successfully rejuvenated cells from elderly donors, some over 100 years old, thus demonstrating the reversibility of the cellular aging process. And good evening, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 5:58 p.m. Eastern Time. Quick note. To achieve this, they used an adapted strategy that consisted of reprogramming cells using a specific cocktail of 6 genetic factors while erasing signs of aging. The researchers provided that the iPSC cells thus obtained then had the capacity to reform all types of human cells. They have the physiological characteristics of young cells, both from the perspective of their proliferative capacity and their cellular metabolisms. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:01 p.m. Eastern Time. Quick note. Using this new cocktail of 6 factors, the senescent cells reprogrammed into functional iPSC cells required the characteristic of embryonic pluripotent stem cells. Good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 6:05 p.m. Eastern Time. Quick note. In particular, they recovered their capacity for self-renewal and their form or differentiation potential. They do not preserve any traces of previous aging. To check the rejuvenated characteristics of these cells, the researchers tested the reverse process. The rejuvenated iPSC cells were again differentiated to adult cells and compared to the original old cells, as well as to those obtained using human embryonic pluripotent stem cells, HESC. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:08 p.m. Eastern Time. Quick now. Signs of aging were erased, and the iPSC cells obtained can produce functional cells of any type with an increased proliferation capacity of longevity. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:09 p.m. Eastern Time. Quick note. Tested on the cells taken from donors over the age of 100. The results obtained led to the research team to test the cocktail on even older cells taken from donors of 92, 94, 96, and even up to 101 years old. Our strategy worked on cells taken from donors in their hundreds. The age of cells is definitely not a reprogramming barrier. This research paves the way for their therapeutical use of iPS, insofar as an ideal source of adult cells is provided, which are tolerated by the immune system and repair organs or tissues in elderly patients. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 6:56 p.m. Eastern Time. Quick note. Brain parasite directly alters brain chemistry. Science Daily, November 4, 2011. Research shows infection by the brain parasite Toxoplasma gondii, found in 10 to 20% of the UK's population, directly affects the production of dopamine, a key chemical messenger in the brain. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 6:57 p.m. Eastern Time. Quick note. Faculty of Biological Sciences believes that the findings could ultimately shed new light on treating human neurological disorders that are dopamine-related, such as schizophrenia, attention deficit hyperactivity disorder, and Parkinson's disease. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 6:58 p.m. Eastern Time. Quick note. This research may explain how these parasites remarkably manipulate rodents. Behavioral for their own advantage. Infected mice and rats lose their innate fear of cats, increasing the chances of being caught and eaten, which enables the parasite to return to its main host to complete its life cycle. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:59 p.m. Eastern Time. Quick note. Stanley Medical Research Institute and Dunhill Medical Trust. The research team found that the parasite causes production and release of many times the normal amount of dopamine in infected brain cells. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:01 p.m. Eastern Time. Quick note. Dopamine is a natural chemical which relays messages in the brain-controlling aspects of movement, cognition, and behavior. It helps control the brain's reward and pleasure centers and regulates emotional responses such as fear. The presence of a certain kind of dopamine receptor is also associated with sensation seeking, whereas dopamine deficiency in humans results in Parkinson's disease. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 7:01 p.m. Eastern Time. Quick note. Based on these analyses, it was clear that T. gondii can orchestrate a significant increase in dopamine production in neural cells. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:06 p.m. Eastern Time. Quick note. Humans are accidental hosts to T. gondii, and the parasite could end up anywhere in the brain. So human symptoms of toxoplasmosis infection may depend on where a parasite ends up. This may explain the observed statistical link between incidences of schizophrenia and toxoplasmosis infection. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:07 p.m. Eastern Time. Quick note. Next experiments will investigate how the parasite enzyme triggers dopamine production and how this may change behavior. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:08 p.m. Eastern Time. Quick note. Toxoplasmosis, which is transmitted via cat feces found on unwashed vegetables and raw or undercooked infected meat, is relatively common, with 10 to 20% of UK population and 22% of the US population estimated to carry the parasite as cyst. Most people with the parasite are healthy, but for those who are immune suppressed, and particularly for pregnant women, there are significant health risks that can occasionally be fatal. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:12 p.m. Eastern Time. Quick note. The parasite infects the brain by forming a cyst within its cells and produces an enzyme called tyrosine hydroxylase, which is needed to make dopamine. Dopamine's role in mood, sociability, attention, motivation, and sleep patterns are well documented, and schizophrenia has long been associated with dopamine, which is the target of all current schizophrenia drugs on the market. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:13 p.m. Eastern Time. Quick note. The enzyme tyrosine hydroxylase is a crucial step in making L-DOPA, prescribed as levodopa for Parkinson's disease, a chemical that is readily converted to the neurotransmitter dopamine. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:17 p.m. Eastern Time. Quick note. Tropical forests fertilized by nitrogen air pollution, scientists find. Science Daily, November 3, 2011. Scientists braved ticks and a tiger to discover how human activities have perturbed the nitrogen cycle in tropical forests. Studies at the remote Smithsonian Institute Global Earth Observatory sites in Panama and Thailand show the first evidence of long-term effects of nitrogen pollution in tropical trees. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 7:23 p.m. Eastern Time. Quick note. Air pollution is fertilizing tropical forests with one of the most important nutrients for growth. Tropical Research Institute in Panama. We compared nitrogen in leaves from dried specimens collected in 1968 with nitrogen in samples of new leaves collected in 2007. Leaf nitrogen concentration in the proportion of heavy to light nitrogen isotopes increased in the last 40 years, just as they did in another experiment when we applied fertilizer to the forest floor. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:26 p.m. Eastern Time. Quick note. Nitrogen is an element created in stars under high temperatures and pressures. Under normal conditions, it is a colorless, odorless gas that does not readily react with other substances. Air consists of more than 75% nitrogen, but nitrogen also plays a big role in life as an essential component of proteins. When nitrogen gas is zapped by lightning or absorbed by soil bacteria called nitrogen fixers, it is converted into other active forms that can be used by animals and plants. Humans fix nitrogen by the Haber process, which converts nitrogen gas into ammonia, now a principal ingredient in fertilizers. Today, nitrogen fixation by humans has approximately doubled the amount of reactive nitrogen emitted. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:30 p.m. Eastern Time. Quick note. Nitrogen comes in two forms, or isotopes, atoms that have the same number of protons but different numbers of neutrons. In the case of nitrogen, the isotopes are 14N and 15N, although only about 1 in 300 nitrogen atoms is the heavier form. Imagine nitrogen in an ecosystem like a bowl of popcorn. Normally the ratio of popped light to unpopped heavy kernels stays the same, but when someone starts to eat the popcorn, the lighter the popped kernels get used up first, increasing the ratio of heavy to light kernels, or 15N divided by 14N in the case of the ecosystem. Light nitrogen is lost through nitrate leaching and as gases such as N2 and the various forms of nitrous oxides or noxides, some of which can be important greenhouse gases. In the fertilization study in Panama mentioned earlier, N2O emissions were tripled. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:36 p.m. Eastern Time. Quick note. Tree rings provide a handy timeline for measuring changes in wood nitrogen content. From the Institute of Botany at the University of Natural Resources and Life Science, who faced down a tiger when sampling trees in the monsoon forest on the Thailand border. We find that over the last century there is an increase in the heavier form of nitrogen over the lighter form, which tells us that there is more nitrogen going into the system and higher losses. We also got the same result in an earlier study of tree rings in Brazilian rainforest. So it looks like nitrogen fixed by humans now affects some of the most remote areas in the world. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 7:39 p.m. Eastern Time. Quick note. The results have a number of important implications. The most obvious is for trees in the bean family, Fabaceae, a major group in tropical forests that fix their own nitrogen association with soil bacteria. Increased nitrogen from outside could take away their competitive advantage and make them less common, changing the composition of tree communities. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:41 p.m. Eastern Time. Quick note. There are also implications for global change models, which are beginning to include nitrogen availability as a factor, affecting the response of plants to increasing atmospheric carbon dioxide concentrations. Most models assume that higher nitrogen equals more plant growth, which could remove carbon from the atmosphere and offset future warming. However, a challenge for the models is that there is no evidence that trees are growing faster in Panama, despite the long-term increases in nitrogen deposition and atmospheric carbon dioxide. Good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:43 p.m. Eastern Time. Quick note. Decades of atmospheric nitrogen deposition have caused major changes in the plants and soils of temperate forests in the U.S. and Europe. Whether tropical forest will face similar consequences is an important question for future research. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:45 a.m. Eastern Time. Quick note. The success of the project to create a reprogrammable cell could revolutionize synthetic biology and would pave the way for scientists to create completely new and useful forms of life using a relatively hassle-free approach. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:46 a.m. Eastern Time. Quick note. We are looking at creating a cell's equivalent to a computer operating system in such a way that a given group of cells could be seamlessly reprogrammed to perform any function without needing to modify its hardware. And Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 8, 2011, 8:47 a.m. Eastern Time. Quick note. We are talking about a highly ambitious goal leading to a fundamental breakthrough that will ultimately allow us to rapidly prototype, implement, and deploy living entities that are completely new and do not appear in nature, adapting them so they perform new useful functions. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 8, 2011, 10:23 p.m. Eastern Time. Quick note. Sea life must swim faster to survive. To survive climate change. Science Daily, November 7, 2011. Fish and other sea creatures will have to travel large distances to survive climate change, international marine scientists have warned. And good evening, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:23 p.m. Eastern Time. Quick note. Sea life, particularly in the Indian Ocean, the Western and Eastern Pacific, and the subarctic oceans, will face growing pressures to adapt or relocate to escape extinction, according to a new study by an international team of scientists published in the Journal of Science. And good evening, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:25 p.m. Eastern Time. Quick note. Our research shows that species which cannot adapt to the increasingly warm waters they will encounter under climate change will have to swim farther and faster to find a new home. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:25 p.m. Eastern Time. Quick note. Using 50 years data of global temperature changes since the 1960s, the researchers analyzed the shifting climates and seasonal patterns on land and in the oceans to understand how this will affect life in both over the coming century. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:26 p.m. Eastern Time. Quick note. We examine the velocity of climate change, the geographical shifts of temperature bands over time, and the shift in seasonal temperatures for both land and sea. We found both measures were higher for the ocean at certain latitudes than on land, despite the fact that the oceans tend to warm more slowly than air over the land. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:27 p.m. Eastern Time. Quick note. The finding has serious implications, especially for marine biodiversity hotspots, such as the famous Coral Triangle and reefs that flourish in equilateral seas, and for life in polar seas, which will come under rising pressure from other species moving in. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 8, 2011, 10:28 p.m. Eastern Time. Quick note. Unlike land-dwelling animals, which can just move up a mountain to find a cooler place to live, a sea creature may have to migrate several hundred kilometers to find a new home where the water temperature, seasonal conditions, and food supply all suit it. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:28 p.m. Eastern Time. Quick note. Under current global warming, the land animals and plants are migrating polewards at a rate of about 6 kilometers a decade, but sea creatures may have to move several times faster to keep in touch with the water temperature and conditions that best suit them. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:30 p.m. Eastern Time. Quick note. We have been underestimating the likely impact of climate change on the oceans. As a general rule, it seems sea life will have to move a lot faster and farther to keep up with temperature shifts in the oceans. This applies especially to fish and marine animals living in the equilateral and subarctic seas, and poses a particular issue both for conservation and fishery management. There is also a complex mosaic of responses globally related to local warming and cooling. For example, our analysts suggest that life in many areas in the Southern Ocean could move northward. However, as a rule, they are likely to be as great or greater in the sea than on land, as a result of its more uniform temperature distribution. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:33 p.m. Eastern Time. Quick note. The migration is likely to be particularly pronounced among marine species living at or near the sea surface, or subsisting on marine plants and plankton that require sunlight, and less so in the deep oceans. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:33 p.m. Eastern Time. Quick note. Also, as seas around the equator warm more quickly and sea life migrates away north or south in search of cooler water, it isn't clear that, if anything, will replace it. No communities of organisms from even warmer regions currently exist to replace those moving out. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:34 p.m. Eastern Time. Quick note. At the same time, sea life living close to the poles could find itself overwhelmed by marine migrants moving in from warmer regions in search of cooler water. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:37 p.m. Eastern Time. Quick note. Castles in the desert satellites reveal lost cities in Libya. Science Daily, November 7, 2011. Satellite imagery has uncovered new evidence of a lost civilization of Sahara Libya's southwestern desert waste that will help rewrite the history of the country. The fall of Gaddafi has opened the way for archaeologists to explore the country pre-Islamic heritage so long ignored under his regime. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:40 p.m. Eastern Time. Quick note. New role for hormone of love, oxytocin in the brain. Helps direct development of brain-body interface. Science Daily, November 2nd, 2011. Much of the body's chemistry is controlled by the brain, from blood pressure to appetite to food metabolism. Structure of one crucial brain area in which biochemical commands are passed from the brain cells to the bloodstream and from there to the body. In the process, the discovery a surprising new role for the hormone of love, showing that it helps to direct the development of this brain structure. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:42 p.m. Eastern Time. Quick note. The area in question, the neurohypophysis, is an interface between nerve fibers and blood vessel located at the base of the brain. Here, some of the major brain-body interactions take place. Hormones released from nerves into the blood vessels regulate a series of vital body processes, including the balance of fluids and uterine contractions in childbirth. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:43 p.m. Eastern Time. Quick note. Although the neurohypophysis has been studied for more than a century, the scientists in the Weizmann Institute led the study developed new genetic tools that enable them to examine the exact three-dimensional arrangement of this brain structure and clarify the cellular and molecular processes leading to its formation. Since the human neurohypophysis is exceedingly complex, the scientists performed the research on live embryos of zebrafish. These fully transparent embryos offer a unique model for studying the vertebrate brain, leading themselves to genetic manipulation with relative ease, enabling researchers to observe the actual formation of a neurohypophysis under a microscope. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:44 p.m. Eastern Time. Quick note. The study revealed a surprising new function for the hormonal messenger oxytocin, dubbed hormone of love because, in addition to controlling appetite, such female reproductive behaviors and breastfeeding, it is also involved mother-child and mate bonding. The scientists showed that oxytocin, one of the two major hormones secreted in the adult neurohypophysis, is involved in the development of the brain area already in the embryo. At this stage, the oxytocin governs the formation of new blood vessels. The messenger helps to build the road for transmitting its own future messages. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:46 p.m. Eastern Time. Quick note. These findings provide an important advance in basic research because they shed light on fundamental brain processes, but in the future they might also be relevant to the treatment of disease. Since the neurohypophysis is one of the only few portions of the brain able to regenerate after injury, an understanding of how it is formed may one day help achieve such regeneration in other parts of the central nervous system. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:15 a.m. Eastern Time. Quick note. Farm fish fed vegetable matter may have residual pesticides. Science Daily, November 8, 2011. Today, half of all the fish we eat comes from fish farms. The problem is that these fish are increasingly being fed vegetable matter, which could lead to a buildup of residual pesticides in them. A new test shows how high the risk of contamination really is. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:17 a.m. Eastern Time. Quick note. People are eating more and more fish. To meet rising demand, fish are increasingly being cultivated in fish farms. Aquaculture is the fastest developing branch of the global food industry, with annual growth of 9%. This rapid rise is also pushing up demand for fish feed, producers of which are facing a formidable challenge. Because the supply of fish meal and fish oil, important ingredients in feed, is dwindling, they are set to be replaced by crops such as soya, maize, and rape. But the trouble with these is that feed pellets made from them might contain pesticides. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:19 a.m. Eastern Time. Quick note. To protect consumers, there has to be a way to test fish bred in captivity for pesticide residues, but this has not been possible up till now. While techniques known as the metabolism studies are already used to test how the active ingredients in pesticides accumulate and break down in ruminants, poultry and pigs, these techniques are not suitable for use with fish. Germany's Federal Office Protection and Food Safety has let it be known that urgent action is required. Now scientists at the Institute of Molecular Biology and Applied Ecology have developed a system to test whether chemical substances accumulate in fish that are fed contaminated feed. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:20 a.m. Eastern Time. Quick note. First, we test whether ingestion of the food feeds leads to a buildup of pesticide residues in fish tissue. Then we look to see which degeneration products or metabolites results from the fish metabolite processes. Essentially, the more fat-soluble a substance is, the higher the probability of it accumulating in fish. Our test from the basis for later studies on feeding the results determine whether these subsequent studies which ascertain maximum pesticide residue levels are required. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:25 a.m. Eastern Time. Quick note. For their metabolism studies, the researchers use water tanks that are 2 cubic meters in size. Into these tanks, they place carp and rainbow trout, each weighing 300 to 500 grams. Both these freshwater fish are frequently bred in farms to detect and identify pesticide residues and their metabolites. A powerful filtering system prevents the dissolved test substance from accumulating in the water. The researchers then test the fresh of these fish for pesticide residues using highly sensitive analytical methods which permit even the smallest quantities of a substance to be detected with certainty. Our new test leaves no stone unturned in the search for pesticides and their degradation products in fish, from breeding through the tissue analysts in the laboratory. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:27 a.m. Eastern Time. Quick note. In autumn 2011, the European Commission will publish new data requirements for fish as part of the approval process for pesticides. These will oblige every producer and importer who intends to bring a new pesticide onto the European market not only to register it, but also to provide information proving it cannot accumulate in the edible parts of fish. The fish test developed at the IME will supply the information required. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:38 a.m. Eastern Time. Quick note. 1. You are keenly interested in studying the metabolism of aromatic amino acids in Escherichia coli, particularly the synthesis of phenylalanine. To initiate your investigation, you decide to isolate phenylalanine oxotrophs by exposing wild-type E. coli to a mutagen. An oxotroph is a mutant that cannot grow on media lacking certain essential molecules that are normally synthesized by the wild-type strain. Hence, in this case, phenylalanine oxotroph would require phenylalanine to be present in the medium in order to grow. A mutagen is a chemical or physical agent that interacts with DNA and causes a mutagen to occur. And good morning, it's Dr. Will, CBMD, PhD. This Wednesday, November 9, 2011, 12:50 a.m. Eastern Time. Quick note. Equations and concepts. The cross product of any two vectors, A and B, is a vector C whose magnitude is given by AB sin degrees and whose direction is perpendicular to the plane formed by A and B. The sense of C can be determined from the right-hand rule. C equals A times B. C in bars equals AB sin degrees. And good morning, it's Dr. Will, CBMD, PhD. This is Wednesday, November 9, 2011, 12:51 a.m. Eastern Time. Quick note. The angular momentum of a particle whose linear momentum is p and whose vector position is r is defined as L r times p. The SI unit of an angular momentum is kg times m divided by s to the power 2. Note that both the magnitude and direction of L depends on the choice of origin. L r times p. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:52 a.m. Eastern Time. Quick note. The torque acting on a particle whose vector position is r can be expressed as r times F, where F is the external force acting on a particle. Torque also depends on the choice of the origin and has an SI unit of N times M. T equals R times F equals N. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:54 a.m. Eastern Time. Quick note. If the same origin is used to define L and T, then the torque on the particle equals the time rate of change of its angular momentum. This expression is the rotational analog Newton's second law, F equals dp divided by dt, and its basic equation for treating rotating rigid bodies and rotating particles, T equals dl divided by dt. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:56 a.m. Eastern Time. Quick note. The angular momentum of a system of particles is obtained by taking the vector sum of the individual angular momenta about some point in the inertial frame. The individual momenta may change with time, which can change the total angular momentum. However, the total angular momentum of a system will only change if a net external torque acts on the system. In fact, the net torque acting on a system of particles equals the time rate of a change of total angular momentum. Infinity T EXT equals DL divided by DT N. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:57 a.m. Eastern Time. Quick note. The angular momentum of a rigid body in the form of a plane lamina rotating about a fixed axis is given by the product IW, where I is the momentum of inertia. About the axis of rotation, W is the angular velocity. L equals IW N. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:59 a.m. Eastern Time. Quick note. Taking the time derivative and using, we find that the net external torque acting on a rigid body is proportional to the angular acceleration alpha. Hence, if the external torque is zero, the body has no angular acceleration. In this case, the body either rotates with constant angular velocity or is at rest. Infinity T EXT equals IA alpha. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:01 a.m. Eastern Time. Quick note. The law of conservation of angular momentum states that if the resultant external torque acting on a system is zero, the total angular momentum is constant. This follows. If infinity T EXT equals DL divided by DT equals zero, L is constant. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:03 a.m. Eastern Time. Quick note. If we apply this result to a body rotating about a fixed axis and the moment of inertia changes from I sub I to I sub F, the conservation of angular momentum can be used to find the final angular velocity in terms of the initial angular velocity. I sub I, W sub I equals I sub F, W sub F equals constant N. Good morning, it's Dr. Will, CBMD, PhD. This is Wednesday, November 9, 2011, 1:04 a.m. Eastern Time. Quick note. If a uniform body of circular cross-section rolls on a rough surface without slipping, the velocity and acceleration of the center of mass are simply related to the angular velocity and angular acceleration according. V sub C equals RW, A sub C equals R alpha. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:05 a.m. Eastern Time. Quick note. The total kinetic energy of a rigid body rolling on a rough surface can be expressed as the sum of the rotational kinetic energy about the center of mass and the translational kinetic energy of the center of mass. K equals 1/2 I sub C W to the power of 2 plus 1/2 MV sub C to the power of 2 N. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:06 a.m. Eastern Time. Quick note. If a body rolls down an incline without slipping, one can use conservation of energy to find the velocity of the center of mass as the body falls through a vertical distance h starting from rest. The result is given. From this expression, one can also find the acceleration of the center of mass. V sub C equals, in bracket, 2gh divided by 1 plus 1 over c divided by mr2, in bracket, closed, 1/2 n. Good morning, it's Dr. Will, CBMD, PhD. This Wednesday, November 9, 2011, 2:49 a.m. Eastern Time. Quick note. If you do not understand interferometry, it is basically the same system used in some laser systems to produce holograms and spectrum analysis. I'm only dealing with generating harmonics and subharmonics for research only. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 2:50 a.m. Eastern Time. Quick note. Rice's microscope may have worked on the principles of using interferometry, and this could be why. To this day, the microscope is not understood. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:01 a.m. Eastern Time. Quick note. Rice's first machine used two transmitters. These two transmitters produce a mixing of frequencies, F1-F2, and the addition of some frequencies, F1 plus F2. In the mixing process, many waves are produced with many fundamentals. The scope pictures above show the mixing process produces. And good morning, it's Dr. Will, CBMD, PhD. It is Wednesday, November 9, 2011, 3:03 a.m. Eastern Time. Quick note. These wideband radio waves must contain all the fundamental frequencies for the known virus. The transmitters are then adjusted to produce the correct fundamental frequency. This is done by adjusting the amplitude output of one of the fundamental oscillators, which change the fundamental frequency and creates many harmonics of other fundamental frequencies, one behind the other, slightly out of phase. An example of this would be to take the 62 Hz at 3 volt peak to peak and 1.059 kilohertz at 5V peak to peak, and mix the two to get a total output of 1.5 volt peak to peak, getting the oscillator at 150 MV, and then modulating the 1.6000 MHz transmitter. The frequency ends up at 1.556120, the fundamentals with all the harmonics added. can you see? N. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:06 a.m. Eastern Time. Quick note. More than 75% of the organisms Rife could see with this universal microscope are only visible with ultraviolet light. But ultraviolet light is outside the range of human vision. It is invisible to us. Rife overcame this limitation by heterodiming, a technique which became popular in the early radio broadcasting. He illuminated the microbe, usually a virus, bacteria, with two different wavelengths of the same ultraviolet light frequency, which resonated with the spectral signature of the microbe. These two wavelengths produced interference where they merged. The interference was in effect a third longer wave, which fell into the visible portion of the electromagnetic spectrum. This was how Rife made invisible microbes visible without killing them, a feat which today's electron microscope cannot duplicate. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:16 a.m. Eastern Time. Quick note. In the expression for torque T equals R times F, is R equal to the moment arm? Explain. Answer: no, the vector R is the position vector of the applied force relative to some origin. The moment arm is a projection of R along the direction perpendicular to F, whose magnitude is the perpendicular distance to the line of action of F from the axis of rotation. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:17 a.m. Eastern Time. Quick note. Can a particle moving in a straight line have non-zero angular momentum? Answer: no. A particle has angular momentum about an origin when moving in a straight line, as long as the line of motion does not pass through the origin. Its angular momentum is zero. If the line of motion passes through the origin, in which case R is parallel to V, and L equals R times P equals R times MV equals 0. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:18 a.m. Eastern Time. Quick note. If the torque acting on a particle about an arbitrary origin is 0, what can you say about the angular momentum about that origin? Answer: since T equals dl divided by dt equals 0, we conclude that L is constant to the motion. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:19 a.m. Eastern Time. Quick note. A particle moves in a straight line, and you are told that the torque acting is 0 about some unspecified origin. Does this necessarily imply that the net force on the particle is 0? Can you conclude that its velocity is constant? Answer: no, the net force is not necessarily 0. If the line of action of the net force passes through the origin, the net torque about the origin is 0, yet F sub net equals 0, therefore you cannot conclude that its velocity is constant. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:21 a.m. Eastern Time. Quick note. Suppose that the velocity vector of the particle is completely specified. What can you conclude about the direction of its angular momentum vector with respect to the direction of motion? Answer: since L equals R times P, L must be perpendicular to the direction of motion. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:23 a.m. Eastern Time. Quick note. If the net torque acting on a rigid body is non-zero about some origin, is there any other origin about which the net torque is 0? Answer: yes, if the origin is one for which the moment arm is 0. The net torque will be 0 about that origin. In fact, any origin for which R times F equals 0, that is R parallel to F, will satisfy this condition. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:24 a.m. Eastern Time. Quick note. If a system of particles is in motion, is it possible for the total angular momentum to be 0 about some origin? Yes, the vector sum of the individual angular momentum can be 0. For example, the two identical particles shown at the right moving in circular orbits, but in the opposite sense, have angular momentum which cancel each other. L sub 1 points out of the page while L sub 2 points into the page. Also, L sub 1 equals L sub 2 equals MVR. Decimal. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:25 a.m. Eastern Time. Quick note. A ball is thrown in such a way that it does not spin about its own axis. Does this mean that the angular momentum is 0 about an arbitrary origin? No, angular momentum is defined as L equals R times P equals R times MV. As you can see from the diagram, the particle has non-zero angular momentum about an arbitrary origin, even when the particle moves in a straight line. L equals 0 only when R is parallel to V, or when V equals 0. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:27 a.m. Eastern Time. Quick note. When a cylinder rolls on a horizontal surface, are there any points on the cylinder that have only a vertical component of velocity at some instant? If so, where are they? No, the diagram shows that every point has a horizontal component equal to R sub W, where R is the radius of the cylinder However, points along a vertical line through the center of mass have only a horizontal component of velocity. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 4:59 a.m. Eastern Time. Quick note. Drinking water from plastic pipes is harmful. Science Daily, November 8, 2011. Pipe and pipe systems are now commonly used to distribute water in many Norwegian homes. The inner pipe for drinking water is made from a plastic called cross-linked polyethylene, PEX. Are these pipes harmful to health, and do they affect the taste and odor of drinking water? And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:01 a.m. Eastern Time. Quick note. Previous international studies have shown that plastic pipes can release substances that give an unwanted taste and odor to drinking water. It has also been suggested that some of these substances may be carcinogenic. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:02 a.m. Eastern Time. Quick note. The aim of the study by the Norwegian Institute of Public Health was to investigate whether leakage products from these pipes are harmful to health and if they affect the taste and odor of drinking water. These leakage products consist of residues of additives used during production to give plastic pipes their desired properties, as well as any subsequent breakdown products. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:03 a.m. Eastern Time. Quick note. The study showed there are no health risks associated with drinking water from PEX pipes. A few types of PEX pipe may cause prolonged undesirable taste and odor if the water remains in pipes over time. Although the taste and odor usually dissipate with use, water from 2 of the PEX pipes still have had an unpleasant smell and taste after a year. The level of volatile organic compounds that leaked from the new PEX pipes was generally low. The level was further reduced with use. No correlation was found between production method and leaking products. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:04 a.m. Eastern Time. Quick note. 10 different types of PEX pipes available in the Norwegian market were tested for leaching products in the standardized laboratory test. The water was in contact with the tubes for 72 hours. 3 different manufacturing methods produce pipes known as PEX A and PEX B or PEX C. These methods use slightly different additives, but this study found no correlation between production method and leakage products. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:05 a.m. Eastern Time. Quick note. 2,4-di-tert-butyl phenol, methyl-tert-butyl ether, MTBE, were 2 of the most commonly occurring substances detected in the water in the experiments. For 3 types of new pipe, MTBE was detected in higher concentrations than the U.S. government's recommended limits for taste and odor of drinking water, U.S. EPA, but the values were reduced to below this limit after the tubes had been in use for a while. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5, 2011, 12:16 p.m. Eastern Time. Quick note. Fruit fly intestine may hold secret a fountain of youth. Long-lived fruit flies offer clues to slowing human aging and fighting diseases. Science Daily, November 2, 2011. One of the few reliable ways to extend an organism's lifespan, be it a fruit fly or a mouse, is to restrict calorie intake. Now, a new study in fruit flies is helping to explain why such minimal diets are linked to longevity and offering clues to the effects of aging on stem cell behavior. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5, 2011, 12:17 p.m. Eastern Time. Quick note. Scientists at the Salk Institute for Biological Studies and their collaborators found that tweaking a gene known as PGC-1, which is also found in human DNA and the intestinal stem cells of fruit flies, delayed the aging of their intestine and extended their lifespan by as much as 50%. And good afternoon, this is Dr. Will, CBMD, PhD. Saturday, November 5, 2011, 12:17 p.m. Eastern Time. Quick note. Fruit flies and humans have a lot more in common than most people think. An associate professor at Salk's Laboratory of Genetics, a lead scientist on the project, there is a tremendous amount of similarity between a human small intestine and the fruit fly intestine. And good afternoon, this is Dr. Will, CBMD, PhD. Saturday, November 5, 2011, 12:18 p.m. Eastern Time. Quick note. Scientists have long known that calorie restriction, the practice of limiting daily food intake, can extend the healthy lifespan of a range of animals. In some studies, animals on restricted diets live more than twice as long on average as those on non-restricted diets. And good afternoon, this is Dr. Will, CBMD, PhD. This is Saturday, November 5, 2011, 12:21 p.m. Eastern Time. Quick note. While little is known about the biological mechanisms underlying this phenomenon, studies have shown that the cells of calorie-restricted animals have greater numbers of energy, generating structures known as mitochondria. In mammals and flies, the PCG-1 gene regulates the number of these cellular power plants. which convert sugars and fats from food into the energy for cellular functions. And good afternoon, this is Dr. Will, CBMD, PhD. This Saturday, November 5th, 2011, 12:22 p.m. Eastern Time. Quick note. This chain of connections between the mitochondria and the longevity inspired Jones and her colleagues to investigate what happens when the PCG-1 gene is forced into overdrive. To do this, they used genetic engineering techniques to boost the activity of the fruit fly equivalent of the PCG-1 gene. The flies, known as Drosophila madagascar, have a short lifespan, allowing the scientists to study aging and longevity in ways that aren't as feasible in longer-lived organisms such as mice or human. And good afternoon, this is Dr. Will, CBMD, PhD. This Saturday, November 5th, 2011, 12:23 p.m. Eastern Time. Quick note. The researchers found that boosting the activity of the DPGC-1, the fruit fly version of the gene, resulted in greater numbers of the mitochondria and more energy production in flies. The same phenomenon is seen in organisms on calorie-restricted diets, when the activity of the gene was accelerated in stem and progenitor cells of this intestine, which serve to replenish intestinal tissues. These cellular changes correspond with better health and longer lifespan. The flies live between 20 and 50% longer, depending on the method and extent to which the activity of the gene was altered. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 12:25 p.m. Eastern Time. Quick note. Part of the reason for this might be that boosting the fruit fly version of PCG-1 stimulates the stem cells that replenish the intestinal tissues, keeping the fly's intestinal healthier. The findings suggest that the fruit fly version of PCG-1 can act as a biological dial for slowing the aging process and might serve as a target for drugs or other therapies to put the brakes on aging and age-related diseases. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:27 p.m. Eastern Time. Quick note. Slowing the aging of a single important organ, in this case the intestine, could have a dramatic effect on overall health and longevity. In a disease that affects multiple tissue, for instance, you might focus on keeping one organ healthy, and to do that, you might be able to utilize PGC-1. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:29 p.m. Eastern Time. Quick note. NASA Fermi finds youngest millisecond pulsar, 100 pulsars today. Science Daily, November 3rd, 2011. An international team of scientists using NASA Fermi Gamma Ray Space Telescope has discovered a surprisingly powerful millisecond pulsar that challenges existing theories about how these objects form. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:33 p.m. Eastern Time. Quick note. A pulsar is a type of neutron star that emits electromagnetic energy at periodic intervals. A neutron star is the closest thing to a black hole that astronomers can observe directly, crashing 500,000 times more mass than Earth into a sphere no larger than a city. This matter is so compressed that even a teaspoonful weighs as much as Mount Everest. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 12:34 p.m. Eastern Time. Quick note. With this new batch of pulsars, Fermi now has detected more than 100, which is an exciting milestone when you consider that before Fermi's launch in 2008, only 7 of them were known to emit gamma rays. And good afternoon, this is Dr. Will, CBMD, PhD. This Saturday, November 5th, 2011, 12:37 p.m. Eastern Time. Quick note. One group of pulsars combines incredible density with extreme rotation. The fastest of these so-called millisecond pulsars whirls at 43,000 revolutions per minute. Millisecond pulsars are thought to achieve such speeds because they are gravitationally bound in binary systems with normal stars. During part of their stellar lives, gas flows from the normal star to the pulsar. Over time, the impact of this falling gas gradually spins up the pulsar rotation. And good afternoon, this is Dr. Will, CBMD, PhD. It is Saturday, November 5th, 2011, 12:39 p.m. Eastern Time. Quick note. The strong magnetic fields and rapid rotation of pulsars cause them to emit powerful beams of energy, from radio waves to gamma rays, because the star is transferring rotational energy to the pulsar. The pulsar spins eventually slows as the star loses matter. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:40 p.m. Eastern Time. Quick note. Typically, millisecond pulsars are around 1 billion years old. However, in November 3rd issue of the Science, the Fermi team reveals a bright, energetic millisecond pulsar only 25 million years old. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:41 p.m. Eastern Time. Quick note. The object name PSR J18233021A lies within an NGC 6624 spherical collection of ancient stars called Globular Cluster, one of about 160 similar objects that orbit our galaxy. The cluster is about 10 billion years old and lies about 27,000 light years away toward the constellation Sagittarius. And good afternoon, it's Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:43 p.m. Eastern Time. Quick note. Fermi's Large Area Telescope, LAT, showed that 11 globular clusters emit gamma rays. The cumulative emission of dozens of millisecond pulsars too faint for even Fermi to detect individually, but that's not the case for NGC 6624. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 12:44 p.m. Eastern Time. Quick note. It is amazing that all of the gamma rays we see from this cluster are coming from a single object. It must have formed recently based on how rapidly it is emitting energy. It is a bit like finding a screaming baby in a quiet retirement home. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 12:47 p.m. Eastern Time. Quick note. J18233021A was previously identified as a pulsar by its radio emission. Yet of the 9 new pulsars, none are millisecond pulsars, and only one was later found to emit radio waves. Despite its sensitivity, Fermi's LAT may detect only one gamma ray for every 100,000 rotations of some of these faint pulsars. Yet new analyst techniques apply to the precise position and arrival time of photons collected by the LAT since 2008 were able to identify them. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 12:51 p.m. Eastern Time. Quick note. Ellen also detects the Einstein Home Project, a distributed computing effort that uses downtime on computers of volunteers to process astronomical data. In July, the project extended the research for gamma-ray pulsars to the general public by including Fermi LAT data and the work processed by Einstein Home users. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 1:06 p.m. Eastern Time. Quick note. Astrobiologists discover sweet spots for the formation of complex organic molecules in the galaxy. Science Daily, November 2nd, 2011. Scientists within the New York Center for Astrobiology Polytechnic Institute have compiled years of research to help locate areas in the outer space that have extreme potential for complex organic molecule formation. The scientists search for methanol, a key ingredient in the synthesis of organic molecules that could lead to life. Their results have implications for determining the origins of molecules that spark life in the oceans. And good afternoon, this is Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 1:07 p.m. Eastern Time. Quick note. Methanol formation is the major chemical pathway to complex organic molecules in interstellar space. In the study director of the NASA-funded center, Douglas, if scientists can identify regions where conditions are right for rich methanol production, they will be better able to understand where and how the complex organic molecules needed to create life are formed. In other words, follow the methanol and you may be able to follow the chemistry that leads to life. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 1:11 p.m. Eastern Time. Quick note. Using powerful telescopes on Earth, scientists have observed large concentrations of simple molecules such as carbon monoxide in the clouds that give birth to new stars. In order to make more complex organic molecules, hydrogen needs to enter the chemical process. The best way for this chemistry to occur is on the surface of tiny dust grains in space. According to, in the right conditions, carbon monoxide on the surface of interstellar dust can react at low temperatures with hydrogen to create methanol, CH3OH. Methanol can serve as an important stepping stone in formation of the much more complex organic molecules that are required to create life. Scientists have known that methanol is out there, but to date there has been limited detail on where it is most readily produced. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 1:13 p.m. Eastern Time. Quick note. Collaborations have discovered is that methanol is most abundant around a very small number of newly formed stars. Not all young stars reach such potential for organic chemistry. In fact, the range in methanol concentration varies from negligible amounts in some regions of the interstellar medium to approximately 30% of the ice around a handful of newly formed stars. They also discovered methanol for the first time in low concentrations, 1 to 2% in the cold clouds that will eventually give birth to new stars. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 1:15 p.m. Eastern Time. Quick note. The scientists conclude in the paper that there is a sweet spot in the physical conditions surrounding some stars that account for a large discrepancy in methanol formation in the galaxy. The complexity of the chemistry depends on how fast certain molecules reach the dust grains surrounding new stars. The rate of molecule accumulation on the particles can result in an organic womb or literal dead end. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 1:16 p.m. Eastern Time. Quick note. This means that under the right conditions, the dust surrounding certain stars could hold greater potential for life than most of its siblings. The presence of high concentrations of methanol could essentially jumpstart the process to create life on the planets formed around certain stars. And good afternoon, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:26 p.m. Eastern Time. Quick note. The scientists also compared their results with methanol concentrations in comets to determine a baseline of methanol production in our own solar system. Comets are time capsules. Comets can preserve the early history of our solar system because they contain material that hasn't changed since the solar system was formed. As such, the scientists could look at the concentrations of methanol in comets to determine the amount of methanol that was in our solar system at its birth. End. Good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:27 p.m. Eastern Time. Quick note. What they found was that methanol concentrations at the birth of our solar system were actually closer to the average of what they saw elsewhere in interstellar space. Methanol concentrations in our solar system were fairly low, at only a few percent, compared to some of the other methanol-dense areas in the galaxy observed. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:28 p.m. Eastern Time. Quick note. This means that our solar system wasn't particularly lucky, and didn't have the large amounts of methanol that we see around some other stars in the galaxy. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:32 p.m. Eastern Time. Quick note. Brains cells responsible for keeping us awake identified. Science Daily, November 3, 2011. Bright light arouses us. Bright light makes it easier to stay awake. Very bright light not only arouses us, but is known to have antidepressant effects. Conversely, dark rooms can make us sleepy. It is the reason some people use masks to make sure light doesn't wake them while they sleep. And good afternoon, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:40 p.m. Eastern Time. Quick note. These cells release a neurotransmitter called hypocretin. The researchers compared mice with and without hypocretin and found that those who didn't have it were unable to stay awake in the light, while those who had it showed intense activation of these cells in the light, but not while they were awake in the dark. And good afternoon, this is Dr. Will, CBMD, PhD. This is Saturday, November 5, 2011, 1:42 p.m. Eastern Time. Quick note. The same UCLA research group earlier determined that the loss of hypocretin was responsible for narcolepsy and the sleepiness associated with Parkinson's disease. What the neurotransmitter's role in normal behavior was until now unclear. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:43 p.m. Eastern Time. Quick note. This current finding explains prior work in humans that found that narcoleptics lack the arousing response to light, unlike other equally sleepy individuals, and that both narcoleptics and Parkinson's patients have an increased tendency to be depressed compared to others with chronic illnesses. And good afternoon, this is Dr. Will, CBMD, PhD. This is Saturday, November 5, 2011, 1:44 p.m. Eastern Time. Quick note. Prior studies of the behavioral role of hypocretin in rodents had examined the neurotransmitter's function during only light phases, normal sleep time for mice, or dark phases, their normal wake time, but not both. And the studies only examined the rodents when they were performing a single task. Good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:46 p.m. Eastern Time. Quick note. In the current study, researchers examined the behavioral capabilities of mice that had their hypocretin genetically knocked out, KO mice, and compared them with the activities of normal, wild-type mice, WT, that still had their hypocretin neurons. Researchers tested the two groups while they performed a variety of tasks during both light and dark phases. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:47 p.m. Eastern Time. Quick note. Surprisingly, they found that the KO mice were only deficient at working for positive rewards during the light phase. During this dark phase, however, these mice learned at the same rate as their WT mates and were completely unimpaired in the working for the same rewards. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:50 p.m. Eastern Time. Quick note. Surprisingly, they found that the KO mice were only deficient at working for positive rewards during the light phase. During the dark phase, however, these mice learned at the same rate as their WT littermates and were completely unimpaired in working for the same rewards. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:51 p.m. Eastern Time. Quick note. Consistent with the data in the KO mice, the activity of hypocretin neurons in their WT littermates was maximized when working for positive rewards during the light phase, but the cells were not activated when performing the same task in the dark phase. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 1:56 p.m. Eastern Time. Quick note. The findings suggest that administering hypocretin and boosting the function of hypocretin cells will increase the light-induced arousal response. Blocking their function by administering hypocretin receptor blockers will reduce this response and thereby induce sleep. Good afternoon, this is Dr. Will, CBMD, PhD. It is Saturday, November 5, 2011, 1:58 p.m. Eastern Time. Quick note. Administration of hypocretin may also have antidepressant properties, and blocking it may increase tendencies toward depression. So we feel this work has implications for treating sleep disorders as well as depression. And good afternoon, this is Dr. Will, CBMD, PhD. It is Saturday, November 5, 2011, 3:17 p.m. Eastern Time. Quick note. Evolution during human colonization, selective advantage of being there first. Science Daily, November 3, 2011. Research published in Science November 3rd reveals that the first individuals settling on the new land are more successful at passing on their genes than those who did not migrate. And good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:19 p.m. Eastern Time. Quick note. We find that families who are at the forefront of a range expansion into new territories had greater reproductive success. In other words, that they had more children, and more children who also had children. As a result, these families made a higher genetic contribution to the contemporary population than those who remained behind in what we call the range core, as opposed to the wavefront. Good evening, it's Dr. Will, CBMD, PhD. It's Saturday, November 5, 2011, 3:21 p.m. Eastern Time. Quick note. The research confirms in humans a phenomenon that has already been observed in other species with much shorter generation spans. We knew that the migration of species into new areas promoted the spread of rare mutations through a phenomenon known as gene surfing. But we find that selection at the wavefront could make the surfing much more efficient. The evolutionary mechanism, in the combination with founder effects and social cultural transmission of reproductive behavior, could explain why some genetic diseases are found at an elevated frequency in the end. Good afternoon, this is Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:22 p.m. Eastern Time. Quick note. It is exciting to see how a study on a regional population of Quebec can bring insight on a human process that has been going on for thousands of years. The Balzac population is a powerful tool for social and genetic research, and this study is a very nice demonstration of its possibilities. And good evening, it's Dr. Will, CBMD, PhD. It's Saturday, November 5, 2011, 3:22 p.m. Eastern Time. Quick note. The researchers also note that although their study concerns a whole human population spread over several centuries, it only represents a short period of human evolution at a limited geographical scale. It thus appears difficult to directly generalize these results obtained in a farmer population to what happened during other range expansions, especially considering the differences between the ecological demography of hunter-gatherer and farmer communities. But given the highly successful history of the human colonization of our planet, it appears very likely that a considerable fraction of our ancestors have lived on the edge of expansion waves. Consequently, several human traits favoring dispersal and reproduction could have evolved during phases of range expansion rather than resulting from selection in constant environments. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:27 p.m. Eastern Time. Quick note. Observations of gamma ray burst reveal surprising ingredients of early galaxies. Science Daily, November 2, 2011. An international team of astronomers led to the Max Planck Institute of Extraterrestrial Physics has used the brief but brilliant light of distant gamma ray burst as a probe to study the makeup of very distant galaxies. Surprisingly, the new observations reveal two galaxies in the young universe that are richer in the heavier chemical elements than the Sun. The two galaxies may be in the process of merging. Such events in the early universe will drive the formation of many new stars and may be the trigger for gamma ray burst. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:28 p.m. Eastern Time. Quick note. Gamma ray bursts are the brightest in explosions in the universe. They are first spotted by orbiting observations that detect initial short bursts of gamma rays. After their positions have been pinned down, they are then immediately studied using large ground-based telescopes that can detect the visible light and infrared afterglows that the bursts emit over the succeeding hours and days. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:31 p.m. Eastern Time. Quick note. One such burst, called the GRB 090323, was first spotted by the NASA Fermi Gamma Ray Space Telescope. Very soon afterward, it was picked up by the X-ray detector on NASA's Swift satellite and with the Grand system at the MPG ESO 2.2 meter telescope in Chile. From Lagrange observations, the astronomers estimated the minimum rate of star formation, which has to be several times higher than the one in our galaxy. They could, however, only determine a minimum value because the detected emission could be heavily deflected, absorbed by the presence of dust in the galaxies. The real rate of star formation, once the unknown dust absorption has been taken into account, could easily be 50 times higher than in the Milky Way. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:45 p.m. Eastern Time. Quick note: the burst was also studied in great detail using ESO, Very Large Telescope, VLT, just one day after it exploded. These observations show that the brilliant light from the gamma ray burst had passed through its own host galaxy and another galaxy nearby. These galaxies are being seen as they were about 12 billion years ago. Such distant galaxies are very rarely caught in the glare of a gamma ray burst. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:47 p.m. Eastern Time. Quick note: when we studied the light from the gamma ray burst, we didn't know what we might find. It was a surprise that the cool gas in these two galaxies in the early universe proved to have such an unexpected chemical makeup. Lead author of the paper describing the new results: these galaxies have more heavily elements than have ever been seen in the galaxy so early in the evolution of the universe. We didn't expect the universe to be so mature, so chemically evolved, so early on. And good evening, it's Dr. Will, CBMD, PhD, this Saturday, November 5, 2011, 3:49 p.m. Eastern Time. Quick note: it is expected that the galaxies in the young universe will be found to contain smaller amounts of heavier elements than galaxies at the present day. Such as the Milky Way, the heavier elements are produced during the lives and deaths of generations of stars, gradually enriching the gas in the galaxies. Astronomers can use the chemical enrichment in galaxies to indicate how far they are through their lives. But the new observation surprisingly revealed that some galaxies were already very rich in heavy elements less than 2 billion years after the Big Bang, something unthinkable until recent. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:56 p.m. Eastern Time. Quick note: the newly discovered pair of young galaxies must be forming new stars at a tremendous rate to enrich the cool gas so strongly and quickly as the two galaxies are close to each other. They may be in the process of merging, which would also provoke star formation when the gas clouds collide. The new results also support the idea that the gamma ray burst may be associated with vigorous massive star formation. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:57 p.m. Eastern Time. Quick note: energetic star formation in galaxies like these might have ceased early on in the history of the universe. 12 billion years later, at the present time, the remains of such galaxies would contain a large number of stellar remnants such as black holes and cool dwarf stars, forming a hard-to-detect population of dead galaxies, just faint shadows of how they were in their brilliant youths. Finding such corpses in the present day would be a challenge. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 3:59 p.m. Eastern Time. Quick note: we were very lucky to observe GRB 090323 when it was still sufficiently bright, so that it was possible to obtain spectacularly detailed observation with the VLT gamma ray burst. Only stay bright for a very short time, and getting good quality data is very hard. We hope to observe these galaxies again in the future. When we have much more sensitive instruments, they would be perfect targets for ELT, concludes. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 4:37 p.m. Eastern Time. Quick note: pulsating response to stress and bacteria discovered. Science Daily, November 3, 2011. If the changing seasons are making it chilly inside your house, you might just turn the heater on. That's a responsible response to a cold environment, switching to a toaster and more comfortable state until it warms up outside. And so it is no surprise that biologists have long thought cells would respond to their environment in a similar way. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 4:40 p.m. Eastern Time. Quick note: but now researchers at the California Institute of Technology, Caltech, are finding that cells can respond using a new kind of pulsating mechanism, instead of just shifting from one steady state to another and staying there. The principles behind this process are surprisingly simple, the researchers say, and could drive other cellular processes, revealing more about how the cells and ultimately life work. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 4:43 p.m. Eastern Time. Quick note: in their environment, the researchers studied how a bacterial species called B. subtilis responds to a stressful environment, for example, one without food. In such conditions, the single-celled organism activates a large set of genes that help it deal with hardship by adding cell repair. For instance, previously biologists had thought the bacteria would handle stress by turning on the relevant genes and simply leaving them on until the stress goes away. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5, 2011, 4:45 p.m. Eastern Time. Quick note: Instead, the researchers found the B subtilis continuously flips these genes on and off. When faced with more stress, it increases the frequency of these pulses. The pulsating action is like switching your heater on full blast for a brief period every few minutes, and turning it on and off more frequently if you want the house to be warm. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 4:48 p.m. Eastern Time. Quick note. To make their finding, the researchers introduced a chemical B subtilis that inhibits the production of ATP, energy-carrying molecules of cells. The team found that the stress induced by this chemical triggers interactions within a set of genes, collectively called the genetic circuit. This circuit, which contains a set of positive and negative feedback loops, generates sustained pulses of activity in a key regulatory protein called sigma B. The researchers attached fluorescent proteins to the circuit, causing the cells to glow green when sigma B was activated. By making movies of the flashing cells, the team could then study the dynamics of the circuit. And good evening, it's Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 4:50 p.m. Eastern Time. Quick note. The key to this pulsating mechanism is the variability inherent in how proteins are made. The researchers say the number of copies of any specific protein in a given cell fluctuates over time. The bacteria gene circuit amplifies these molecular fluctuations, also called noise, to generate discrete pulses of sigma B activation. The stress also activates another key protein that modulates the pulse frequencies. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 4:51 p.m. Eastern Time. Quick note. By turning a steady input, the stress into an oscillating output, the activation of sigma B, the genetic circuit, is analogous to an electrical inverter, a device that converts the direct current DC into alternating current AC. And good evening, it's Dr. Will, CBMD, PhD, this Saturday, November 5th, 2011, 4:52 p.m. Eastern Time. Quick note. This work provides a blueprint for how relatively simple genetic circuits can generate complex and dynamic behaviors in individual cells. The researchers say, we are excited to think that similar mechanisms may occur in other cellular processes. It'd be interesting in the future to see which aspects of this circuit architecture also appear in more complex systems, such as mammalian cells. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 5:07 p.m. Eastern Time. Quick note. Climate shift could leave some marine species homeless. Science Daily, November 3rd, 2011. Rising temperatures will force many species of animals and plants to move to other regions and could leave some marine species with nowhere to go, according to new research just published in the Journal of Science. And good evening, it's Dr. Will, CBMD, PhD. It's Saturday, November 5th, 2011, 5:08 p.m. Eastern Time. Quick note. An international research team led by Dr. Mike Burrows from the Scottish Association for Marine Science compared changing temperatures for both land and sea and from place to place over a 50-year period from 1960 to 2009. And good evening, it's Dr. Will, CBMD, PhD. Saturday, November 5th, 2011, 5:08 p.m. Eastern Time. Quick note. The team used the data to predict how quickly populations of both terrestrial and marine species would have had to relocate to keep up with changing temperatures. They found that there was very little difference between movement rates in either environment. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 5:09 p.m. Eastern Time. Quick note. Dr. Burrows explains when temperatures rise, plants and animals that need a cooler environment move to new regions. The land is warming about 3 times faster than the ocean, so you might simply expect species to move 3 times faster on land, but that's not the case. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 5:12 p.m. Eastern Time. Quick note. If the land temperature becomes too hot for some species, they can move to higher ground where temperatures are generally cooler. That's not an option for many marine species, which live at or near the surface of the ocean. When temperatures rise, species such as fish will be able to move into deeper water to find the cooler environments they prefer, but other species such as marine plants or slow-moving corals will have to move further to find suitable habitats and could become trapped if there are no cooler places for them to go. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 5:15 p.m. Eastern Time. Quick note. Co-author Dr. John Bruno from the University of North Carolina agrees that many marine creatures would have a hard time keeping up with climate change. Being stuck in a warming environment can cause reductions in the growth, reproduction, and survival of ecologically and economically important ocean life, such as fish, corals, and seabirds. And good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 5:17 p.m. Eastern Time. Quick note. The study also highlights the variation in ocean surface temperatures within a very small region, which also causes species movement. Springtime temperatures in the seas around Scotland, for example, have arrived around 5 days per decade earlier on the east coast, whereas there has been almost no shift in spring temperature on the west coast. Good evening, it's Dr. Will, CBMD, PhD, Saturday, November 5th, 2011, 5:18 p.m. Eastern Time. Quick note, Dr. Burrows concludes the areas where a species would need to relocate the fastest to stay ahead of climate changes are important biodiversity hotspots such as the Coral Triangle and southeastern Asia. Our study may help conservationists to prepare for change and protect future coral habitats. Good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:22 a.m. Eastern Time. Quick note, city lights could reveal ET civilizations. Science Daily, November 3, 2011. In their search for extraterrestrial intelligence, astronomers have hunted for radio signals and ultra-short laser pulses. A new paper, Harvard-Smithson Center for Astrophysics and Edwin Turner, Princeton University suggests a new technique for finding aliens. Look for their city lights. Looking for alien cities would be a long shot, but wouldn't require extra resources. And if we succeed, it would change our perception of our place in the universe. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:23 a.m. Eastern Time. Quick note, as with other SETI methods, they rely on the assumption that aliens would use Earth-like technologies. This is responsible because any intelligent life that evolved in the light from its nearest star is likely to have artificial illumination that switches on during the hours of darkness. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:24 a.m. Eastern Time. Quick note, how easy would it be to spot a city on a distant planet? Clearly this light will have to be distinguished from the glare from the parent star. Turner suggests looking at the change in light from an exoplanet as it moves around its star. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:25 a.m. Eastern Time. Quick note, as the planet orbits, it goes through phases similar to those of the moon. When it is in dark phase, more artificial light from the night side would be visible from Earth than reflected light from the day side. So the total flux from a planet with city lighting will vary in a way that is measurably different from a planet that has no artificial lights. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:27 a.m. Eastern Time. Quick note, spotting this tiny signal would require future generations of telescopes. However, the technique could be tested closer to home using objects at the edge of our solar system. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:29 a.m. Eastern Time. Quick note, Turner calculates that today's best telescopes ought to be able to see the light generated by Tokyo-sized metropolis at the distance of Kepler Belt, the region occupied Pluto, Eris, and thousands of smaller icy bodies. So if there are any cities out there, we ought to be able to see them now. By looking, astronomers can hone the technique and be ready to apply it when the first Earth-sized worlds are found around distant stars in our galaxy. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:31 a.m. Eastern Time. Quick note, it is very unlikely that there are alien cities on the edge of our solar system, but the principle of science is to find a method to check. Before Galileo, it was conventional wisdom that heavier objects fall faster than light objects. But he tested the belief and found they actually fall at the same rate. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:32 a.m. Eastern Time. Quick note, as our technology has moved from radio and TV broadcast, it is capable of fiber optics. We have become less detectable to aliens. If the same is true of extraterrestrial civilizations, then artificial lights might be the best way to spot them from afar. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:37 a.m. Eastern Time. Quick note, Hubble directly observes a disk around a black hole. Science Daily, November 4, 2011. A team of scientists has used the NASA ESA Hubble Space Telescope to observe a quasar accretion disk, a brightly glowing disk of matter that is slowly being sucked into its galaxy's central black hole. Their study makes use of a novel technique that uses gravitational lensing to give an immense boost to the power of the telescope. The incredible precision of the method has allowed astronomers to directly measure the disk size and plot the temperature across different parts of the disk. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:39 a.m. Eastern Time. Quick note, an international team of astronomers has used a new technique to study the bright disk of matter surrounding a faraway black hole. Using the NASA ESA Hubble Space Telescope combined with the gravitational lensing effect of stars in a distant galaxy, the team measured the disk size and studied the colors, and hence the temperatures, of different parts of the disk. These observations show a level of precision equivalent to spotting individual grains of sand on the surface of the moon. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:45 a.m. Eastern Time. Quick note, while black holes themselves are invisible, the forces they unleash cause some of the brightest phenomena in the universe. Quasars, short for quasar-stellar objects, are glowing disks of matter that orbit supermassive black holes, heating up and emitting extremely bright radiation as they do so. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 1:45 a.m. Eastern Time. Quick note. A quasar accretion disk has a typical size of a few light days, or around 100 billion kilometers across, but they lie billions of light years away. This means their apparent size, when viewed from Earth, is so small that we will probably never have a telescope powerful enough to see their structure directly. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 1:46 a.m. Eastern Time. Quick note. Until now, the minute apparent size of quasars has meant that most of our knowledge of their inner structure has been based on theoretical extrapolations rather than direct observations. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 1:47 a.m. Eastern Time. Quick note. The team therefore used an innovative method to study the quasar using the stars in an intervening galaxy as a scanning microscope, to probe features in the quasar disk that would otherwise be far too small to see. As these stars move across the light from the quasar, gravitational effect amplify the light from different parts of the quasar, giving detailed color information for a line that crosses through the accretion disk. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 1:48 a.m. Eastern Time. Quick note. The team observed a group of distant quasars that are gravitationally lensed by the chance alignment of other galaxies in the foreground, producing several images of the quasar. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 1:51 a.m. Eastern Time. Quick note. They spotted subtle differences in color between the images and changes in color over time. The observations were carried out. Part of these color differences are caused by the properties of dust in the intervening galaxies. The light coming from each one of the lensed images has followed a different path through the galaxy, so the various colors encapsulated information about the material within the galaxy, measuring the way and extent to which the dust within the galaxies block light, known to the astronomers as the extinction law. At such distances itself, an important result in the study. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 1:53 a.m. Eastern Time. Quick note. For one of the quasar they studied, though there were clear signs that the stars in the intervening galaxy were passing through the path of the light from the quasar, just as the gravitational effect due to the whole intervening galaxy can bend and amplify the quasar light. So can that of the stars within an intervening galaxy subtly bend and amplify the light from different parts of the accretion disk as they pass through the path of the quasar light. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 1:56 a.m. Eastern Time. Quick note. By recording the variation in color, the team were able to reconstruct the color profile across the accretion disk. This is important because the temperature of the accretion disk increases the closer it is to the black hole, and the colors emitted by the hot matter get blurred the hotter they are. This allowed the team to measure the diameter of the disk of hot matter and plot how hot it is at different distances from the center. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:01 a.m. Eastern Time. Quick note. They found that the disk is between 4 and 11 light days, across approximately 100 to 300 kilometers, while the measurement shows large uncertainties. It is still a remarkably accurate measurement for a small object at such a great distance, and the methods hold great potential for increased accuracy in the future. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:01 a.m. Eastern Time. Quick note. This results is very relevant because it implies we are now able to obtain observational data on the structure of these systems, rather than relying on the theory alone. Quasar physical properties are not yet well understood. This new ability to obtain observational measurements is therefore opening a new window to help understand the nature of these objects. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 2:02 a.m. Eastern Time. Quick note. Gravity bends the structure of space-time and hence deflects beams of light when the alignment is right, with one object directly behind another. The foreground objects. Gravity bends the light like a lens, a process called gravitational lensing. Gravitational lens typically produces multiple distorted images of a distant object. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:03 a.m. Eastern Time. Quick note. The most dramatic effect of gravitational lensing are amplification and distortion of light from distant galaxies as it passes through massive galaxy clusters. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:04 a.m. Eastern Time. Quick note. This effect also takes place on a smaller scale, with galaxies at an intermediate distance, lensing the light of distant quasars, producing multiple images of them that are visible through the lens galaxy. And Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:05 a.m. Eastern Time. Quick note. Individual stars can also lens light, although this effect, called gravitational microlensing, is much more subtle and can only be detected by measuring how the lensing effect increases the source's brightness. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:06 a.m. Eastern Time. Quick note. The study makes use of gravitational microlensing by stars in the foreground galaxy to study the accretion disk of quasar in the background. It also uses the interplay of quasar light and gravitational lensing to probe the gas and dust content of intermediate galaxies. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:15 a.m. Eastern Time. Quick note. It takes two brains, comes wired for cooperation, neuroscientists discovered. Science Daily, November 3, 2011. When Nancy Grace and her partner danced a lively rumba, Sponda Ballott's 1980s hit. True on a recent Dancing with the Stars. More going on with the legal commentator's brain than worry over a possible wardrobe malfunction. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 2:22 a.m. Eastern Time. Quick note. In addition to shedding light on ourselves as social and cooperative beings, the results have important implications for engineers who want to be able to program autonomous robots to work effectively as teams in settings such as bomb squads and combat. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 2:24 a.m. Eastern Time. Quick note. Brains among vertebrate animals, frogs, cats, fish, bears, and even humans are more similar than most people realize. The neurotransmitter systems that control brain activity at the molecular level are nearly identical among all vertebrates, and the layout of the brain structures is the same. Thus, the kinds of phenomena that we have described in these wrens is very relevant to the brains of most, if not all, vertebrate species, including us humans. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:24 a.m. Eastern Time. Quick note. Carbon-based organic carriers in interstellar dust clouds, newly discovered diffuse interstellar bands. Science Daily, November 2, 2011. The discovery of 13 diffuse interstellar bands with the longest wavelengths to date could someday solve a 90-year-old mystery. Astronomers have identified the new bands using data collected by Gemini North Telescope of Stars in the center of the Milky Way. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:26 a.m. Eastern Time. Quick note. Nature now reports on its website findings that support recent ideas about the presence of large, possibly carbon-based organic molecules, carriers, hidden in interstellar dust clouds. The paper will also appear in the November 10th print issue of the journal. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:27 a.m. Eastern Time. Quick note. These diffuse interstellar bands, or DIBs, have never been seen before. Spectra of stars have absorption lines because gas and dust along the line of sight of the stars absorb some of the light. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:28 a.m. Eastern Time. Quick note. The most recent ideas are that diffuse interstellar bands are relatively simple carbon-bearing molecules similar to amino acids. Maybe these are amino acid chains in space, which supports the theory that the seeds of life originated in space and rained down on planets. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:29 a.m. Eastern Time. Quick note. Observations in different galactic sight lines indicate that the material responsible for these DIBs survives under different physical conditions of temperature and density. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:30 a.m. Eastern Time. Quick note. The low-energy absorption lines figure and his colleagues discovered provide constraints for determining the nature of diffuse interstellar bands. Future theoretical models that predict wavelengths absorbed by these mysterious particles now must accommodate these lower energies. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:31 a.m. Eastern Time. Quick note. We saw the same absorption lines in the spectra of every star. If we look at the exact wavelength of the features, we can figure out the kind of gas and dust between us and the stars that is absorbing the light. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:32 a.m. Eastern Time. Quick note. Diffuse interstellar bands have remained a puzzle since their initial discovery 90 years ago. The 500 bands identified before the study most occur at visible and near-infrared wavelengths. The observed lines do not match predicted lines of simple molecules and cannot be pinned to a single carrier. And Good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:32 a.m. Eastern Time. Quick note, none of the diffuse interstellar bands has been convincingly identified with a specific element or molecule, and indeed their identification individually and collectively is one of the greatest challenges in astronomical spectroscopy. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:33 a.m. Eastern Time. Quick note, the newly discovered infrared bands can be used as probes of the diffuse interstellar medium, especially in regions in which thick dust and gas obscure observations in optical and shorter wavelength bands. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 4:36 a.m. Eastern Time. Quick note, studying the stronger emissions in the group may lead to an understanding of their molecular origin. Someday laboratory spectroscopy could be used to identify the infrared diffuse interstellar bands. No one has been successful yet at reproducing the interstellar bands in the laboratory, due to the multitude of possibilities and the difficulty of reproducing the temperatures and pressures the gas would experience in space. And